My name is Ashwin Kumar. I'm the sales associate at Strand. Today's presentation is on DNA-Seq data analysis using Avadis NGS. The presenter is Ramesh Hariharan, who is the CTO at Strand. What we hope to achieve today is take you through the workflow for DNA-Seq and Avadis NGS and show you the endpoints that you can reach with the software. If you have any questions throughout the presentation, please don't hesitate to ask. The way to do that is to use the chat feature in Webex, and we'll aim to either answer your question right then or at the end of the presentation. At the end of the presentation, we'll be following up with you individually to learn more about your specific interest in next-generation sequencing, and if you have any plans on the near future in which we can facilitate your research. So with that, here is Ramesh.
Hello everyone, my name is Ramesh Hariharan, and in the next 30 minutes or so I'll lead you through the DNA-Seq features in Avadis NGS. Brief introduction to Avadis NGS. The core design principle behind Avadis NGS is what we call NGS for the rest of us, meaning that the interfaces have been specially designed to hide all the complexity of the large data sets and the complex processing behind fairly visual and easy to use interfaces. So in particular, we've kept the end user biologist in mind while designing these interfaces. And if you're familiar with GeneSpring, Avadis NGS has been developed on the same platform as GeneSpring, which means that you get to see the same user interface. In terms of import, Avadis NGS starts with importing in aligned files. These are files that you take the raw reads that come out of an come out of a sequencer and then align them to the reference genome of interest using one of several alignment tools that are freely available. And all of these tools will typically output a SAM or a BAM file, which is a standard format, output alignment format that we import into Avadis NGS. If you're using the Illumina eLand aligner, then the eLand output text format is also importable into Avadis NGS. And Avadis NGS has 3 different workflows: ChIP-seq, RNA-seq, and DNA-seq. For the rest of this presentation, we'll be focusing only on the DNA-seq part of Avadis NGS. And we'll be seeking to answer the following questions. How do you import your data into Avadis NGS? How do you then QC the reads and get rid of the reads that are not good enough? And once you've done that, if you could either be doing a whole genome experiment, or you could be doing a more targeted resequencing experiment where you're sequencing only targeted regions of the genome, typically through a target enrichment method like SureSelect. So how do you determine the efficacy of this target enrichment? Once you've done that, you're ready to call variants in the genome. So how are SNPs called in Avadis NGS? And then once you have SNP calls for all your samples in an experiment, how do you compare SNPs across samples to find SNPs that are either present in one or not present in one but not in the other, etc. A special case of that, of course, is somatic mutations, where you have tumor normal pairs and you want to identify mutations that have occurred somatically in the cancer sample but are not present in the germline normal sample. And once you have a list of interesting SNPs, how do you determine the effect of these SNPs on the underlying transcripts? And finally, how are structural variants, long insertions, deletions, translocations, inversions, etc. in the genome determined? So let's walk through each of these questions one by one. To begin with, how is data imported? What you see here is the import dialog in Avadis NGS. So the very first thing you have to do is to indicate the organism, the genome build, and the transcript model of interest. We have prepackaged annotations available for a variety of organisms, so for most of the standard organisms and several others as well. From within Avadis NGS, you'll automatically see what we call an annotations manager that will show you all of the available packages for the various organisms. In case you're working with an organism that you don't see on that list, please contact us, and we've been quite active about on-demand supporting new organisms. So once you specify the organism and the build and the transcript model, you have to choose the platform type, which is either Illumina, Life Technology, Solid, 454 Roche, PacBio. We also have support very recently released for the Iron Torrent platform as well. And then once you specify the platform type, you need to specify whether your library protocol was a single end, a paired end, or a mate paired protocol. Using paired end or mate paired data, you can run the structural variant detection algorithm that uses the paired read information, location information to identify insertions, deletions in the genome. If you're working with single end data, which doesn't have, without paired end, then you can still call small variations, namely SNPs and small InDels. So once you've specified all of that information, it's time to specify the input files of interest. As I mentioned earlier, you've got to import in SAM and pre-aligned files, align outside and bring them in as SAM, BAM, or ELIN format files. You can also give a smaller alias to each of your input files so that you can give a name that's more easy to remember. And very often it happens that a single sample gets split into multiple samples, into multiple files as an output of the sequencer or as an output of the alignment. And you can combine multiple files for a particular sample into one single file by giving all of the relevant files the same alias. So in this instance, we've got a blood sample and a tumor sample from the same individual. The tumor sample is split into two different files. The blood sample has a single file. And by giving the same label to both the tumor files, we are essentially combining the reads from both those files into a single sample. So the input to the experiment will be one blood sample and one tumor sample. So now once you specify that and you you run the import process, the files will get imported in, and you'll then get to see the first view of what the user interface looks like, and this is what it looks like. For those of you who are familiar with GeneSpring, you'll see the similarity. The layout and the user interface models that of GeneSpring. I'll walk you through the various elements of this interface. On the left top, you see your work organized into projects and experiments. A project comprises multiple experiments. Each experiment comprises potentially multiple samples that you're trying to analyze together. What you see in the window on the top left is a single project open with a list of experiments. So you could have, for instance, an exome experiment or a whole genome experiment or an RNA-Seq. You could have several of these experiments in the same project, and you could open one or more of these together simultaneously. On the right side, you have what we call application-specific workflows. So for every type of experiment, there's a specific workflow comprising the steps necessary for analyzing data on that experiment. So if you open a ChIP-seq experiment, you'll see a different workflow on the right. If you open an RNA-Seq experiment, you'll see a different workflow on the right. As I mentioned, you can have multiple experiments open, and as so there's a notion of an active experiment. So as you switch the active experiment between the various experiments that are open, the workflow on the right will switch appropriately. the workflow on the right, as you can see, comprises all the steps of starting with setting up the experiment, then doing quality inspection on the reads, filtering out bad reads, doing analysis, SV, structural variant and SNP analysis, and then doing downstream steps relating the variants that you found to genes and their functional impact, so go analysis and pathway analysis. On the left, again, you see various sections, each of which refers to a particular experiment that's open. Within that open experiment, you see a hierarchy of objects. This is the audit trail for that experiment, comprising various objects that you create as you do your analysis. To begin with, after you've just created the experiment, you'll have a single object called the all-aligned reads object, which comprises all of the reads that you've imported in. And as you run various filtering steps and analysis steps, more objects will get created. These objects will either be further read lists, subsets of reads of the all-aligned read lists, or region lists, for instance, SNP lists or lists of structural variants. Any subset of regions on the genome is called a region list. And then as you take variants of interest and identify which genes are impacted by these variants, you'll create gene lists. And then as you take gene lists and ask which pathways are impacted, you'll create pathway objects, hierarchical clustering objects, etc. So all of these reside in the navigator on the left, and the hierarchy reflects the order in which these objects were generated. And finally, the centerpiece is the genome browser in the middle, where you can, which is your window to the data. You can actually see all of your reads, your region lists, your gene lists, the genes and transcripts, DB SNP information, all of the relevant information. You get a view into all of that through the genome browser. Okay, now that we've imported in our samples and we're familiar with the user interface, let's go through the workflow of standard collection of steps that we would run for while going through a DNA-Seq experiment. So the very first step is to QC the reads and identify bad reads and filter these out. There are many ways to look at the quality of the reads, and I'll walk you through a few of these. This view on the top right, the heat map view that you see, is a view that shows you, and this is specific to data coming from the Illumina platform. This shows you on the rows, you see various tiles on which sequencing has been performed, and the columns refer to the base position. So if you've sequenced reads of length 100, then you'll have 100 columns, and the color of a particular cell indicates the quality of the bases in that particular tile. So for instance, the 50th column would indicate the quality of the 50th base in all the reads. And for a particular tile, let's say the 10th row, and the 50th column represents the quality of the 50th bases in all of the reads generated from the 10th tile. Typically, as you go to the right of this heat map, the color turns from red to yellow to blue, indicating that the quality is falling as you go to later flows, meaning to the right end of the read from the left end. But if it so happens that you see a dip to yellow or blue right at the beginning, as is indicated on this bad tile here, so right at the beginning of the read, the color has gone to yellow. It means that tile is bad. The reads that are coming out of that tile have poor base quality, and it's easy to identify visually these tiles and filter all the reads from these tiles out. Likewise, if you look at the scatter plot on your left, this shows on the x-axis, again, you have tiles. So sequencing, every tile contributes a certain number of reads. And this plot shows for each tile how many reads it contributes. But the reads are further segregated based on how well they match in the genome, to the reference genome. If they match with no mismatches, those reads appear in pink. If they match with one mismatch, they appear in red, and so on. You see this one particular tile where there's a sharp dip in the pink and a sharp rise in the red, indicating that perfect match reads, the number of perfect match reads is reduced substantially, and the number of reads which require a mismatch to match the genome has increased substantially. It indicates that there are more errors in this tile than in the other tiles, and so again, this might be a bad tile which is contributing erroneous reads. So you can identify this tile, take all the reads that are generated from this tile and remove all of those reads. Then the pie chart at the bottom shows you the distribution of reads by what we call the mate status. If you're running paired analysis, and then you expect your read pairs to map at a certain expected distance in the genome. So if I have two reads, read 1 and read 2, which together comprise a pair, and my insert length is, let's say, 200, then I expect read 1 and read 2 to map at a distance of 200 in the genome. If they map at a distance larger than 200, then we say that's a case of mate far. If they map at a distance smaller than 200, we say that's a case of mate near. And if only one of the reads maps and the other doesn't, then we say it's a case of mate missing. And if both the reads map and they map at the right expected distance, then we say it's a normal pair. So the proportions of the various read pairs that are normal or mate far or mate missing or mate near are shown in this pie chart. If you see a lot of mate missing reads, that immediately tells you that something went wrong in the alignment, and that you might want to align again with different parameters. If you see very few normal reads and a lot of aberrant reads, aberrant meaning mate near, mate missing, mate far, etc. Again, either something has gone wrong in the alignment or something has gone wrong in the data generation. So that's one set of QCing on reads that you can do. The next set of read QCs has a direct impact on the SNP calls that you'll be making later. And that has to do with what we call alignment scores. So every read, when it maps to the genome, it is also given an alignment score which captures the number of mismatches that are needed to make this read map to the genome. So in your picture here, you see that there's a collection of reads that map to a particular location in the genome, and these are snapshots from our genome browser. In fact, all of the screenshots that you see in this presentation are all taken directly from Avadis NGS. So in this picture, you see that there's a collection of reads, all of which are mapping to this location in the genome, but with a lot of mismatches and gaps. It appears as if the number of mismatches and gaps is way too many to allow for the possibility that some sort of sequencing error or mutation distance between genomes was responsible for this. This is probably just bad mapping. So you can identify reads with low alignment scores, i.e. many mismatches and gaps, and filter those out. And the way you can identify those is we show you a histogram at the bottom right on the distribution of alignment scores. And then you can pick a cutoff and say, remove all reads which have alignment score lower than, let's say, 84, and then it'll remove all of these strange reads with lots and lots of mismatches and gaps. Clearly, if you were not to remove these reads, you'll see that you'll be calling many SNPs and variants within just the span of the reads that are shown on this in this picture, and very likely all of those variants would be erroneous. Likewise, there's another feature that a read has, and that's called its mapping quality. When you mouse over individual reads in the genome browser, it will show you a little report like this, which indicates the mapping quality of a read. The highest mapping quality is 255, and anything that's substantially lower than 255 is poor mapping quality. So what is mapping quality? So a read which has low mapping quality maps at several places in the genome. So the aligner cannot make up its mind on which of these several places does it actually come from, and so it actually ends up depending on the aligner, some aligners will just put this read at all of those places, but assign each of those locations a low mapping quality to tell you that I can't make up my mind where the read comes from, so I'm putting it at all of the locations where I think it could have come from, but I'm assigning to each of those locations a low mapping quality. So a value of 60, which is much less than 255, constitutes low mapping quality. We again show you a distribution, a histogram which shows you the distribution of mapping qualities, and you'll see out here a big spike on the right at 255. So most of the reads have good mapping quality. That's good. And then there are various smaller number of reads at lower numbers between 160 and 10. So these are all the low mapping quality reads. And again, you can set a cutoff and say remove all the reads which have mapping quality lower than 40, let's say. And so you'll retain only reads which are reasonably unambiguously placed in the genome. Again, if this step was not done, then we've observed that it leads to a lot of spurious SNP calls and structural variant calls as well. The last QC on reads that you want to do is to remove duplicates. Here, for instance, you see a collection of reads, all of which have the same sequence and they have the same start and stop location. So these are all reads which are all probably sequencing the same fragment, the same end of the same fragment, and so it's what we call these duplicate reads. The problem with duplicate reads is that they are not independent observations of a particular base, and therefore, if there's an error on the PCR, then that error is going to show up in all of these reads. And what you ideally need for SNP calling is independent reads which assay a particular base independently. So a lot of duplicate reads that is considered unnecessary, and in fact skews the SNP calling algorithm. So we have a duplicate removal that you can specify the number of duplicates you want to retain. Typically 2 or 3 duplicates is about the max that people retain. And all of the other duplicates are removed, and you get a new read list which comprises, which is duplicate free, so to say, or duplicate minimal. So those are all of the QCing steps on the reads that you can perform to get a good set of reads. And if you remember the audit trail hierarchy I showed, as you run these filters to remove reads, you'll create new read lists. And you can then run subsequent operations on any of these read lists. So you can actually experiment with many of these filters and see how many SNPs get called with each filter. Moving on to the next question, how is target enrichment efficacy determined? You can import in a target regions file. So let's say you've run targeted enrichment, you've run target enrichment and then sequencing. So you can take all the targeted regions and import them in as a bed file, and you can drag that into the genome browser, and you'll be able to see those targeted regions and juxtaposed with the transcripts and the reads. In this case, clearly the targeted regions are nicely juxtaposed with exonic regions in the transcript because this is a exome enrichment kit. Now, one of the key questions that you would ask is which of these regions is indeed well sequenced and which of these target regions is not well sequenced. So we have mechanisms in there which will annotate each of these target regions with the number of reads on these target regions. And if you look at the distribution, what you see out here is the distribution of across target regions. And now if you wanted to identify which of my target regions are not well covered enough, you can use the filter on the right out here and say, give me the target regions which have very few reads on them, let's say less than 100 reads on them. And that will create a new region list which comprises only target regions with few reads on them. And that new region list again gets stored in the audit trail navigator. And you can again drag it into the genome browser and view that list. So once you've QC'd the data and you're confident that you've got a good set of reads and that a good chunk of your targets are indeed well covered, and you have a list of targets which are not well covered so that you can assay them separately in some other form. You're ready to call variants now. So how are SNPs called in Avadis NGS? So the SNP calling, Avadis NGS has a built-in SNP calling algorithm that is based on the fairly well-known MAQ or MAC algorithm. The algorithm, very simply speaking, works as follows. You go through every locus in turn, and for the locus at hand, you look at all the reads below that locus and say look at the distributions of the bases on those reads at that locus and pick the two most prevalent bases. So below a locus you've got A's and G's as being most prevalent, and then very few C's and T's. Then you take A and G, those are the two most prevalent bases. And your candidate genotypes at this locus now are AA, AG or GG. So you compute a probability that the underlying genotype is indeed AA or AG or GG. So you do it individually for each of these three cases. And this probability, the input to computing this probability is all of the bases at that locus, and so the base calls at that locus and the associated base qualities. So using the base calls and the base qualities, we compute a probability of the underlying genotype being AA, AG or GG. For instance, if most of the bases are A, clearly the probability of an AA will be high. If most of the bases are G, the probability of a GG will be very high. If the bases are sort of equally or broadly balanced between A and G, then the probability of an AG will be very high. So you've computed three probabilities. You take the largest of these, and instead of reporting it as a probability, we actually convert it to what we call a decibel score, meaning you take it to the -10 log base 10 scale. This scale ensures that the larger the decibel score, it's higher the probability. It's a greater confidence in the call. So if the score is greater than a specified cutoff, you call a SNP with the associated genotype. By associated, I mean that if the probability of AA was the highest, then you say that there's an AA at that location. So that's the algorithm. Of course, the meat is in the probability calculation, and that's done using the MAQ method. So I won't go into further detail here, but if anybody is interested, we can send you more material on this. This is what the SNP dialog looks like. As you can see, there are more parameters that you can provide the algorithm. So in addition to the decibel score cutoff, which as I mentioned, you calculate the decibel score, which is a transformation of the probability, and higher the decibel score, the greater your confidence in the SNP. And you have a cutoff which by default is 50, which so by default that's 50, and so it'll only report SNPs with a decibel score greater than 50. You can set it to a different threshold if you want. But more importantly, there are these other filters that you can also combine with the probability calculation. So you can say that I'm interested only in loci where there are at least 10 overlapping reads. So if you see fewer than 10 reads, don't even bother doing this probability calculation. And there should be at least, of the 10, there should be at least 2 variant reads, as far as the reference is concerned. So the base in these reads is different from the base in the reference. If you're doing 454 analysis, then there are also homopolymer factors that one can bring in to ensure that the SNPs that you're finding are not close to homopolymer stretches. So once you go through the algorithm, it will create for you a region list comprising all of the locations in the genome which have a variant SNP call. And then you can drag that into the genome browser and you can view it. So here, for instance, in the genome browser, we're showing you a homozygous SNP. The indications for that, you can see each of the reads, and the reference has a G and the reads are all A's, so that's clearly a homozygous variant SNP. The color coding bar at the top indicates the homozygous SNP. If it were heterozygous, then you'll see the bar having 2 distinct colors. The Avadis NGS SNP call clearly all A's, so therefore an AA homozygous call. the associated DB SNP. This SNP is known in DB SNP, and the associated DB SNP ID is shown here. And you can also optionally bring in very easily important new tracks, let's say a repeat regions track, to see which of these SNPs lie in repeat regions, and there are simple ways to then filter out SNPs which do not lie in repeat regions. Even though I talked about SNP calling, what we are calling is actually a more general class of variations, which includes small indels, multiple nucleotide polymorphisms, and in addition to SNPs. So here, for instance, is a deletion. You see all of the reads have the T missing, and so the Genome browser shows that with a dash, a hyphen in each of the reads. Here you see an insertion. Between the A and the T, there's a little character. If you mouse over that character, it will tell you what the insert sequence is. It's an A, and it's indicated by these violet little bars that are there. And likewise, out here you see a multiple nucleotide polymorphism. That's two or more variations that come together, in which case we combine them and report combined genotype calls for combining all of those locations. In addition to the genome browser, you can also view SNPs in what we call a variant support view, and I'll tell you why that is interesting. So you pick a SNP in the table on the top, so that's the selected SNP, and note that it's a GT heterozygous call. So at the bottom, you see the variant support view for that SNP, and what the variant support view does is that it takes all of the reads below that locus and it clusters them. So here you have cluster 0 that has 12 reads in it. The size of the cluster is at the extreme right. It has 12 reads in it, and all of these reads have a G in the locus of interest, and the rest of the surrounding loci, they match the reference. So a dot indicates that it matches the reference. And then cluster 1 has 11 reads, and it matches the reference at the locus of interest, so it has a T at the SNP locus. So you have cluster 0, 12 reads with a G and 11 reads with a T. So what the variant support view allows you to do is to get a very compact view of the variation around a particular locus, which would be a little hard if you had like 50 reads, but if you compress those 50 reads into 2 or 3 clusters, it becomes a lot easier to see. So that's how SNPs are called. That's how SNPs can then be verified. Once you call your list of SNPs, you put it in the genome browser and the variant support view and verify and gain confidence that indeed the calls are correct. Once you've done that, you're ready to compare SNPs across samples. And your tool to do that is what we call the Multi-Sample SNP Report. So when you run SNP analysis, it will create a comprehensive table for all the samples in your experiment with the SNP calls across all of these samples. So this multi-sample report integrates SNPs across all samples. So let's take an example here. So you've got an 80 SNP call in the tumor sample, and the corresponding call in the normal sample has this label called ref-1. Ref-1 simply indicates that it's the same as reference, so that normal has there's no SNP call on the normal, it's same as reference. In the tumor, there's an 80 SNP call. Now, from the perspective of filtering out SNPs, if you wanted to say pick SNPs that are present in tumor but not in normal, you can use the filter on the right side out here to specify, you could say normal equals ref-1 or normal does not equal ref-1, or normal equals ref-1 and tumor does not equal ref-1, and so on. So you could specify filter conditions to just filter out SNPs which are present in normal but not in tumor, etc. This piece of functionality, we expect it to be augmented substantially in the future as we add more and more sorts of comparisons across samples. For the moment, there are some sorts of comparisons that you can do right away, and others could be done simply by exporting out this table, and then you could write small scripts to do specific comparisons of your own interest. You can also filter out as you can see, this table also shows novel or dbSNP indicators, which SNPs are known in dbSNP and which are not. So if you wanted to filter out SNPs based on novelty, based on chromosomal location, etc., all of those can be done using these filters. Let's talk about a specific type of comparison across samples, so namely somatic mutations, for which we have special support. The goal of somatic mutation finding is to find SNPs that are present in tumor but not in normal or vice versa. Now, the reason we've introduced a special algorithm to do this is that if you were to just do the filtering on SNP calls that I described just now on the multi-sample report, we found that this yields many false positives because of cutoff issues, namely that you have a SNP in the tumor sample clearly, and in the normal sample the SNP is not being called, but when you go and look at that location in the genome browser or the variant support view, you see that the SNP could be present, but it's the number of reads which are supporting that SNP is somehow much smaller in the normal sample than the tumor sample, but it's clearly not a slam dunk case of the SNP being not present in the normal. So to avoid this problem, to get around this problem, we instead look for differences in base distributions between the tumor and the normal sample. So you look at the AGCT distribution in the tumor sample, the AGCT distribution in the normal sample at that locus, and if the frequency distributions are substantially different, then you say that this is a potential somatic mutation location where the tumor and the normal are behaving differently. And these differences are quantified using a particular statistical test called the Jensen-Shannon divergence test. And again, you can take the outputs and drag it into the genome browser and view these SNPs. And you'll see two sorts of behaviors on the output. One sort is where, so on the top you see the normal sample, on the bottom you see the tumor sample. And we are focusing on a locus which has been output by the somatic mutation finding algorithm as being different in base distributions between the normal and the tumor sample. So in this case, you see that at the top in the normal sample you have your mix of colors. There's less red and more orange, and at the bottom in the tumor sample there's more red and less orange. So it indicates that the mix of bases has changed substantially between tumor and normal. And the other sort of output is the one on the right, where you see clearly that there is a mutation in the tumor that's just not present at all in the normal. So both these sorts of mutations are output by the somatic mutation finding algorithm. Next, let's look at how is the effect of a SNP on transcripts determined. So once you have your list of SNPs, you can do what we call SNP effect analysis. And what that would do is it will go through every one of the SNPs in your input region list, and it will look at each of the overlapping transcripts, and it will look at their reading frames and see what change happens to the reading frames. A number of effects are predicted. So the list of effects that we predict is shown on the right. So clearly the effect on the reading frame could be a non-synonymous change, it could be a synonymous change, it could cause a change in the frame itself, so frame shift change. It could be an intronic SNP. It could be upstream or downstream of the gene itself, and so on. So all of those effects are predicted, and a comprehensive spreadsheet created which has all of this information, including the exon number where the change occurs and the amino acid change in case it's a non-synonymous coding SNP, and so on. So you can, from a SNP prioritization perspective, you can call SNP effects and then pick up SNPs that have the most prominent effects. So you could pick up SNPs which have non-synonymous changes or stop gained or stop loss changes, and prioritize those for further work. Finally, let's look at how long insertions and deletions are and more complicated variants like translocations and inversions are determined. The structural variant finding algorithms, Avadis NGS has a built-in structural variant finding algorithm, and that algorithm works using paired end data, and it works on the following principles. So let's look at how a deletion is found. So for instance, here we have a reference genome in which the blue section which is not present in the subject's genome that you're assaying. So if you have paired reads, the two red reads out here, they form a pair, and you see that these reads have an orientation that I haven't talked about in the past. So this orientation determines the strand that they come from. So these reads have an orientation, and depending on the platform you're using, let's say you're using the Illumina platform, then one of the reads will be what we call a forward read, the other will be a reverse read, so they'll be pointing towards each other in a pair. And the distance between these two reads is expected to be, it's a known insert length distance, because in the assay you've selected for fragments which are in a tight insert length band. So on the subject's genome, let's say you expect these two reads to come from, let's say, 200 base pairs of each other. Now when you map these reads to the reference, because there's an intervening blue region that was deleted off in the subject's genome, these two reads will map at a so that the mutual distance is actually much more than 200, much more than it was in the subject's genome. And the fact that their mutual distance is much more than the expected 200 signifies that there's a big deletion happening here. So we look for read pairs which are further apart than expected, and based on that call long deletions. Likewise, the reads which map further away in the reference indicate a deletion. Likewise, let's look at a case of an insertion. You have the reference genome, and now it's the subject genome's turn to have a blue region, so that's an insertion in the subject genome. And now you'll see that if you have two reads that map at a certain distance length, let's say 200 in the subject genome, because the blue region is not present in the reference genome, these two will now map to being much closer to each other than the stipulated length of 200. So reads which map closer together in the reference indicate an insertion. Now there are more complicated patterns that you can discover, and I'll walk you through a couple of these. You could discover inversions. So you have a reference genome which has a blue region, and that region is just turned around, so it's pointing in the wrong direction in the subject genome. So it's pointing to the right in the reference, but it's pointing to the left in the subject genome. So it's just a flip of that region. It's in the same place where it should be, but it's just flipped around. So that's called an inversion. Now what happens to the reads? So imagine two pairs of reads, the red pair and the brown pair. Each of the pairs, the reads within each of the pairs map within the stipulated, are within the stipulated length of 200 on the subject genome. But once you map these reads to the reference genome, you see what goes wrong. So the red read on the leftmost red read maps to the corresponding red read here. But its mate out here is now mapping all the way to the right end of this blue region because of the inversion. The read was generated from the left end of the blue region in the subject genome, but because of the inversion, it maps to the right end of that region in the reference genome. So two things have happened. So for the red pair, you see that two things have happened. One is that the reads are now further apart than they were in the subject genome. That's number one. But number two, the orientations of the red read pair in the subject genome, they were pointing towards each other. In the reference genome, they're actually pointing in the same direction. And likewise for the brown read pair. So there's a change in distance. There's also a change in orientation. And by detecting this concomitant change in distance and orientation, we can actually identify inversions. So the reads map out of orientation and further away from each other, indicating an inversion. Likewise, you can find large insertions. In a large insertion in the subject, there's a big region that's inserted. What would happen is that you have these two reads from the red pair. One of those will map to the reference. The other doesn't have a place to map at all because it's in the inserted region, which is missing from the reference. So here you would have a case of mate missing, meaning that one read in a read pair matches the reference, the other does not map to the reference at all. So the other, since it doesn't map to the reference at all, we say that the red read pair is a mate missing pair. So reads which have mate missing in the reference indicate a large insertion. And likewise, you can identify translocations. The picture does get a little more complicated. If you have this blue inverted, so this is an inverted translocation to add to the complexity. You'll see that the red read pair actually now maps further out, and it maps with both reads in the same orientation in the reference. Likewise for the brown, but look at the relative placements of the red and the brown. The brown is now nested within the red, which wasn't the case in an inversion, a plain inversion where the brown and the red were interleaved. In an inverted translocation, the brown is now nested within the red. So by looking at patterns of how read pairs behave from a mutual distance and orientation perspective, we can actually call out all of these structural variants, and here are a couple of examples. So this is an example of a homozygous deletion. You can see that, so in our genome browser, the dotted lines are used to connect read pairs. So if you have two reads which are both coming from the same pair, they are connected by a dotted line. So you can see that there's a collection of reads which have a very long dotted line connection compared to all the other reads which have very short dotted line connections out here. So that indicates a big deletion. And likewise, here is an insertion where these pink reads have much shorter dotted line connections than the other gray reads, which are the normal reads. So to reiterate, using paired end information, using the orientation of read pairs and using their mutual distance, one can call all kinds of fairly complex rearrangements in the genome. So that brings me to the end of the feature description on the DNA-Seq side that we wanted to talk about. If any of this piques your curiosity or you have DNA-Seq data that you want to try out on Avadis NGS, how can you try out Avadis NGS? Simply go to our website and click on the Start Evaluation, and that will get you a 15-day trial that you can download and use for free. The website is www.avadisngs.com. On the website, you'll see Avadis NGS installers for Mac, Windows, and Linux. Minimum requirements for hardware are you should check those before you download the installer of interest. If you wanted to learn and get a feel for usage, you can our webinars will typically be archived online, so you can watch these webinars. There's also some amount of demo data sets and demo material, learning material on the website, along with other resources like annotations and pathways, and a number of scripts. So scripting is our way to dynamically enhance functionality of Avadis NGS. So if you use Avadis NGS and you realize that there's a function that you'd like which is not present in the package, you can get in touch with us and request a script. And we've been quite good again about honoring these requests to whatever extent possible. So a number of scripts that we've written for customers have now found their way into the website. Here you'll see a number of scripts that we've put up. So you could download one of these scripts and run it, and this will give you added functionality. So monitor the script library and all the annotations and pathways and demo data sets, all of these resources on the website. With that, we come to the end. Thank you for attending the webinar, and please, we'll get into the question session, so please feel free to ask any questions or clarification that you might have.